muscle. You know where in the body those muscles are found. We went over the major functions of muscle last time. We talked about some of those properties that are unique to muscle tissue. We talked as well about the relationship between connective tissue and that of skeletal muscle. We looked at that. We talked about nerve supply. We talked about blood supply. We mentioned a few things about the embryology of skeletal tissue as well. We also started looking at the individual muscle fibers. We talked about those muscle fibers. We talked about the composition of the membrane of muscle cells. We talked about those invaginations, those T-tubules. We mentioned the idea that when it comes to the filaments that we find, that there's two distinct filaments, those that are thick and thin. We talked about how they're going to be composed of different proteins. All of you recall now the importance of the sarcoplasmic reticulum and how useful it is for calcium storage and for the release of calcium when it comes time for muscle contraction. We mentioned a few things last time about atrophy. We talked about hypertrophy. We talked and we looked at the arrangement of the filaments that we have in the functional unit of muscle, that of the sacromere. We saw the electron micrograph of the sacromere, zooming in at 20,000 times to see those distinct patterns that we have in the sacromere. From one Z-disc to another makes up the sacromere. We talked about DOMS, delayed onset muscle soreness, last time, and that microscopic damage done to the muscle. We also mentioned some of those proteins that muscle is composed of. The contractile proteins, myosin and actin, the regulatory proteins, troponin and trypomyosin, and then all the other proteins that are useful for maintaining alignment with the thick and thin filaments. We looked at the thick filaments, the two golf club type appearance that have the shafts twisted together. We looked at that arrangement of the thick and the thin filament. We saw that there was some overlap between the thick and the thin filament here in the A band in the zone of overlap. We mentioned some of those other structural proteins that maintain the alignment. We know when it comes to muscle contraction that there is no crinkling up of these filaments, but that these filaments are going to pass on top of one another, that they slide past one another. And we started talking a little bit about contraction last time. We talked about how we need a nerve impulse to course down a neuron, reaching the terminal portion of the neuron, the synaptic end bulb. It's there where we find our neurotransmitter. When it comes to skeletal muscle contraction, the neurotransmitter of importance is what? Acetylcholine. Good. Yes. Acetylcholine binds to receptor sites on the membrane of the muscle. Postsynaptically is where we find it. So that acetylcholine diffuses across the synapse to those receptor sites. When acetylcholine binds to acetylcholine receptors, that causes sodium channels to open, and we have an influx of sodium from the extracellular fluid. This, as you know, as we talked about, due to positive feedback, causes more sodium channels to open and more, and we have propagation of that potential along the membrane of the muscle. It's coursing over the surface of the sarcolemma, down into those invaginations, down into those T-tubules, where we have a very intimate relationship with the sarcoplasmic reticulum. It is that impulse that causes the release of calcium from the sarcoplasmic reticulum, and calcium binds to troponin, and it causes troponin and trypomyosin to move out of the way to allow myosin to bind to actin. And this is what starts the contraction cycle. So we can see here that arrangement of the calcium within the sarcoplasmic reticulum. When the action potential courses down the muscle, we see the release of that calcium, and we see the binding site for myosin now being exposed when calcium is present. When it comes to the contraction cycle, there are a few repetitive steps in the contraction cycle, hence the idea of a cycle. And so long as we have certain criteria met, i.e., enough calcium and enough energy in the form of ATP, these steps will continue, where we will hydrolyze ATP, we attach myosin to actin, and it forms a cross bridge. We then need to ratchet forward, this maneuver known as the power stroke, and then we need to be able to repeat this process. So actin and myosin need to detach from one another. We want to lock that we need to lock that head back, and we can repeat. And as I said, so long as we have energy in the form of ATP and a supply of calcium available, this contraction cycle can continue. And it looks something like this. When it comes to our myosin head, it is going to be responsible for hydrolyzing ATP. So what happens? We're going to cleave off that terminal phosphate group and be left with ADP plus our phosphate. This is going to reorientate that of the head, and the head is going to bind to that binding site on the thin filament, causing that of our cross bridge being formed. After the cross bridge is formed, we have a ratcheting type motion where this myosin head is going to rotate toward the middle of the sarcomere, and this is the power stroke, as these filaments are literally sliding past one another. After we slide past the filament, we then need to detach that head, and we need to be able to repeat this process. So we are going to detach, we're going to use another molecule of ATP, and this process will repeat. It repeats so long as we have those binding sites exposed, thanks to the presence of calcium, and a ready supply of ATP available. Questions at all? Well then, let me check with my online students to see if they have any questions.